Welcome to this lecture on the general characters of Cnidarians. If you have ever been to the beach and seen a jellyfish or marveled at a coral reef in a documentary, you have already encountered this incredible group of animals. Today, we are going to explore what makes them so unique. We will start from their basic biological structure, understand their ecological importance, and even dive into the fascinating physics behind their microscopic spring-loaded weapons. By the end of this session, you will see these simple creatures in a completely new light. Let us begin by imagining you are swimming in the ocean and suddenly feel a sharp sting, even though nothing bit you. You have just met a Cnidarian. This phylum includes familiar animals like jellyfish, sea anemones, and corals. You can think of a Cnidarian as a living, floating water balloon that is heavily armed with microscopic, spring-loaded harpoons. What makes them truly fascinating is what they lack. They do not have brains, they do not have bones, and they do not have a heart. Yet, despite lacking these complex organs, they are some of the most effective and ancient predators on our planet. Studying them gives us a direct window into the early days of animal evolution. Now that we have a general idea of what these creatures are, let us look at their formal biological definition. Cnidarians are aquatic, mostly marine, invertebrate animals. Unlike simpler organisms like sponges, Cnidarians have reached a tissue level of biological organization, meaning their cells work together in coordinated ways. A key defining feature is their radial symmetry. Imagine a bicycle wheel with spokes radiating from the center. A Cnidarian's body is organized around a central axis in exactly the same way. This evolutionary design is brilliant for a floating or attached animal because it allows them to interact with their environment, sense danger, and capture food from all directions equally. Building on this radial symmetry, let us examine how their bodies are actually constructed. Cnidarians are described as diploblastic. The prefix di means two, which tells us that their bodies are built from only two true cellular layers. The outer layer, which interacts with the water, is the epidermis, also known as the ectoderm. The inner layer, which lines their gut, is the gastrodermis, or the endoderm. But what keeps these two layers together? Sandwiched between them is a jelly-like non-cellular substance called the mesoglia. This mesoglia acts as a flexible skeleton, giving jellyfish their characteristic squishy, gelatinous form while supporting their overall structure. Beyond their body layers, we must understand how they process food and defend themselves. Instead of a complex digestive tract like ours, Cnidarians possess a single central space called a gastrovascular cavity. Interestingly, this cavity has only one opening. This single opening must serve as both the mouth to take in food and the anus to expel waste. But how do they catch this food? The entire phylum gets its name, Cnidaria, from their most defining feature, specialized stinging cells called cnidocytes. Inside these cnidocytes are explosive, microscopic organelles known as nematocysts. These are the tiny harpoons used for capturing prey and defending against predators. To truly grasp how this specialized stinging cell works, let us look at a detailed diagram of its mechanism. On the left, you see the resting state of the cnidocyte. Notice the small trigger hair at the top called the cnidocyl. Inside the cell, protected by a lid called the operculum, sits the venom capsule containing a tightly coiled thread. Now, look at the right side, showing the discharged state. When prey brushes against the cnidocyl, the operculum snaps open. In a fraction of a second, the coiled thread shoots outward, turning inside out to expose sharp barbs. These barbs anchor into the prey, allowing the venom to be injected. It is a perfect microscopic trap. Now that we understand their biology and their deadly microscopic weapons, you might wonder why these creatures matter to us. Cnidarians are incredibly important. Reef-building corals, which are cnidarians, are the architects of the ocean. They create massive ecosystems that support roughly 25% of all marine life. Without them, entire oceanic food webs would collapse, devastating global fisheries. Beyond ecology, they have revolutionized medicine. The green fluorescent protein, originally discovered in jellyfish, is now a vital tool used worldwide to track cancer cells and study genetic diseases. Furthermore, scientists are heavily researching their unique venoms to develop highly effective new painkillers and neurological drugs. Having seen their massive ecological and medical impact, let us return to their most fascinating feature, the nematocyst. To truly appreciate this evolutionary marvel, we must look at the physics behind it. When the cnidocyte is triggered by physical touch or chemical signals, the harpoon fires with astonishing speed. In fact, this is one of the fastest cellular processes in the entire animal kingdom, happening literally in the blink of an eye. This explosive discharge is driven by intense, built-up osmotic pressure inside the cell capsule. Even though this is a biological event, we can perfectly understand the mechanical power behind it by looking at basic kinematic equations from physics. To quantify this explosive discharge, we rely on a fundamental principle of kinematics. The formula on screen shows how to calculate average acceleration. Conceptually, acceleration measures how quickly an object changes its speed over a specific period of time. Let us break down the symbols. The letter A represents the average acceleration, measured in meters per second squared. The variable VF stands for the final velocity of the barb right before it strikes, while VI is the initial velocity. Since the harpoon starts at rest, VI is 0. Finally, T represents the incredibly tiny amount of time the discharge takes, measured in seconds. Because this time T is in the denominator and is extremely small, the resulting acceleration becomes massively large. Now that we have our formula, let us apply it to a real biological scenario. A researcher observes a hydra firing a nematocyst. We are given that the microscopic harpoon reaches a final velocity of 2 meters per second. We are also told this happens in just 2 microseconds, which is 0.0000002 seconds. Since the harpoon is initially coiled inside the cell, its initial velocity is 0. We substitute these values into our acceleration formula. We subtract 0 from 2 in the numerator and divide by our tiny time value in the denominator. The result is an average acceleration of 1 million meters per second squared. This mind-boggling number is roughly 100,000 times greater than Earth's gravity. Knowing the acceleration is just the first step. This brings us to the actual physical force exerted on the prey. To find this, we use Sir Isaac Newton's second law of motion. Conceptually, this law states that the force an object carries depends on both how heavy it is and how fast it is accelerating. In our formula, F represents the force, measured in newtons. The variable m is the mass of the object in kilograms, and a is the acceleration we just discussed. This formula reveals a crucial principle. Even if an object like our microscopic harpoon has a vanishingly small mass, it can still generate a deadly significant force if its acceleration is pushed high enough. With Newton's second law in hand, let us calculate the exact striking force of this microscopic weapon. We are given the mass of the harpoon, which is an incredibly small 0.000000005 kilograms, equivalent to half a microgram. We also have our massive acceleration of 1 million meters per second squared from the previous calculation. We substitute these known values into our force equation, multiplying the tiny mass by the huge acceleration. The result is a force of 0.0005 newtons. While half a millinewton sounds tiny to us, remember, it is applied over the razor-sharp tip of a harpoon. This creates immense localized pressure that easily shatters the exoskeletons of microscopic prey. We have covered a remarkable journey from basic biology to biomechanical physics. Let us review our key takeaways. First, we learned that the phylum Cnidaria represents a crucial stepping stone in evolution, introducing tissue-level organization and radial symmetry. Second, despite their simple diploblastic bodies, they are highly successful predators. This success is entirely due to their specialized cnidocytes and the explosive nematocysts hidden inside. Third, by applying kinematic physics, we proved that extreme biomechanical acceleration allows these microscopic harpoons to generate enough force to pierce biological armor. Finally, never forget their global importance. These simple creatures build massive coral reefs and provide invaluable resources for modern scientific and medical research. You have now mastered the general characters of Cnidarians.